Good day, ladies and gentlemen. My name is Marshall Slabbert and I'm a master's student from the Northwest University. And today I will be presenting my research project, which is the biodiversity of the endophytic fungi and bacteria associated with sainfoin on Obricus vicifolia in South Africa. Now, maize is one of the most important primary food sources in South Africa, and thus the increased production thereof is vital to ensure food security. However, factors such as plant parasitic nematodes, for example Legionella brasilensis and Mallory, can severely limit the production and yield of maize, thereby decreasing food security. So in this context, sainfoin, Obricus vicifolia, which is a perennial forage legume, may have the potential to suppress plant parasitic nematode populations due to the production of anthelmintic condensed tannins in its leaves and stems. So ultimately, more research regarding sainfoin is thus needed. For example, its root endophytic biodiversity, which has yet to be studied, to investigate its agricultural potential and also enable higher introduction of sainfoin into maize-based agricultural systems. So the aim of this study was thus to evaluate the biodiversity of the endophytic fungi and bacteria associated with sainfoin roots in South African agricultural soils. And the objectives were to isolate and identify endophytic fungi and bacteria from the roots of sainfoin during the wet and dry seasons, and then to compare the biodiversity of the obtained isolates between seasons to see if there is a variation. And lastly, a literature-based evaluation of the potential plant growth promoting effects of the obtained isolates. So the methods included sainfoin sampling during the wet and dry season, and this was done on an established agricultural field on a farm called Jakkelsfontein near Vicksburg, South Africa, followed by isolation, culturing, and purification of the root endophytic fungi and bacteria, and then DNA-based identification of the isolates, which entailed DNA extraction, PCR, and sequencing, followed by phylogenetic analysis, and lastly, a literature-based evaluation of the plant growth promoting effects of the obtained isolates. Now, thus far, 10 bacterial and 15 fungal isolates have been obtained during the dry season, which are in the process of identification. And for the wet season, 24 bacterial and 28 fungal isolates have been obtained, which are also in the process of identification. Now, it is expected that sainfoin will have a high endophytic microbial diversity that shows seasonal variation with a higher biodiversity during the wet season, and also includes bacterial and fungal species with plant growth promoting effects that can be applied in agriculture. Thank you.